Okay, so we're gonna start chapter 7. I'm probably gonna let you guys leave a little early today. We're running well ahead of schedule, which will also based on our syllabus, and I'll give you guys a chance to maybe absorb a little material instead of our crash courses. Does that sound good to you guys? Okay. So let's go to our PowerPoint here. We are going to do special senses, and by that I mean special sensory organs. So what are we specifically going to be looking at today? We're going to be looking at like eyes, hearing, these guys right here. These are our specialty senses. Not all of these have their own organ. So we have smell, olfaction, gustation, which is taste, vision, equilibrium, and hearing. These guys all go together. These are all part of the ear apparatus. So we're basically going to do nose, tongue, eyes, and our ears. So we're going to be kind of looking at 4 big areas for our special sense organs. And today we'll probably get through olfaction and gustation. We'll see about vision. Everybody good? All right, so what is olfaction? It is our sense of smell. Which cranial nerve's in charge of our sense of smell? Cranial nerve what? Oh, to touch and feel. It's our first, this is cranial nerve number 1, is our olfactory bulbs, right? So you were close. 1, 3, no. All right, so it's cranial nerve number 1 does our olfaction. Remember how on the brain, you guys are like, we are done with that practical. We don't have to think about it anymore, right? But if I was looking at the underside of my brain, we had our olfactory bulb up here like this, right? So this was, and we numbered from 1 down to 12 like this. So that's cranial nerve number 1. It does your sense of smell. So we learned that, to touch and feel. And then we said some say, so the some stood for our sensory. It is a pure sensory nerve. So out here on our olfactory bulbs, we're going to be talking about how do we take an odor in the air and turn it into a smell that our brain registers as a smell. Interesting thing about the sense of smell is it is the only nerve, this nerve right here, is the only nerve that directly connects to the outside world. We have them hanging down in our nose. What's the bone that the olfactory nerves come through? Do you guys remember from 201? Ethmoid bone. And they go through what little holes in the ethmoid bone? Yeah, the cribriform foramen or olfactory foramen. So we had our ethmoid bone. We have little holes. If we go sideways, I'm just going to kind of draw my guy's little face here. Here's his mouth. Here's his nose right here. Here's his eyeball. Your nerves, your olfactory bulbs come out to the front of your brain like this, and the nerves actually hang right down into the top of your nose like this. They are covered with a protective tissue. What's our known protective tissue? Yeah, but what kind of cells? Our ET, epithelial cells, right? These are our protectors. So these nerves hang right down into the top of our nose, and they have an epithelial cell layer over them, which protects them a bit. It is a mucous membrane. I think someone mentioned that we secrete mucus. So it's a moistened epithelial tissue that actually covers up these free hanging nerve endings that are hanging right down in the top of our nose. So when we began to look at our sense of smell, we're going to look at the olfactory epithelium, which is kind of covering up our free nerve endings that are hanging right down exposed in our nose. And our lamina propria. What is lamina propria? Do you remember from 201? Yeah, it's the proper layer. It's the bottom. So all epithelial tissues sit on what? No, well, these are columnar ones, I sure know. But if these are epithelial tissues, they're avascular, right? So can they free float and survive? No, because they don't have blood supply. So who do they have to be sitting on? Connective tissue. This connection where my epithelial tissues join my connective tissue is my lamina propria. So it's kind of this anchoring base layer is our lamina propria. All right, so we have our neurons. They're highly modified nerve cells. Like I said, the most unique nerve cells in that they have direct contact with the outside world. How do we send a signal? All right, do you guys remember back to 201 when we talked about action potentials? How did we kick off an action potential? All right, let's backtrack to cell membranes. So if I have my cell membrane and it's at resting state, if this is my cell membrane, this is outside my cell, inside my cell, and it's at resting potential, you have to think about your sodium potassium pumps, right? You guys learn about sodium potassium pumps. Where am I pumping sodium? Yes. Oh, you're pointing. Yes, you're right. Sodium goes out here outside my cell. And who goes inside my cell? Potassium. And I create a concentration gradient, right? Now, how did I start an action potential? How did I depolarize? What is polar? Water. Well, water is polar, but what does polar mean? It has a charge to it, right? I had an unequal charge. Why? Because these are ions. Ions are charged. What does that mean? At some point, these guys were partnered up with some other molecule or formed a molecule, and then they broke up. And when they broke up, they didn't give their stuff back. And so these guys have a positive charge. So they were attached to something that kept their electrons. So they have more protons than electrons, but it gives them a charge, right? These guys would be positively charged ions. Sodium and potassium are positively charged ions, but they don't have the same charge. So when I pump my sodium out and my potassium in, do you remember we had about a -70 difference in voltage? How did we send an action potential rippling down the cell? What was the first thing that happened? My class better know this because I drew this like a million times for you on the board, right? Channel open? Yeah, the channel open. What causes the channel to open? Well, sodium goes in when the channel opens, but what causes this gate to open? What? Chemicals, that's one thing. What else can open my gates? There's three big things that can open our gates. Chemicals, charge, that size. This one we didn't talk about. I talked about just enough to name it, but we didn't really get into the logistics. But it's mechanical distortion. What does that mean? If I distort my membrane, it'll open the gate. When it opens the gate and sodium flows in, it starts my action potential, right? So there are three ways I can open and send an action potential. I can open my gate using a chemical. I can open my gate based on voltage. We saw that here, right? Our potassium channels. As the voltage changed on the inside of the cell, it opened this and closed this, right? You guys remember that? Those are voltage gates. So I can open and close these guys using changes in charge or mechanical distortion. All right, so when we get into our sense of smell, what's going to open our gate? It's going to be a chemical. How do we get the chemical into the gate, which are going to send our action potential? They absorb into my epithelial tissues. For them to absorb in, we want to have some moisture to my epithelial tissues. So we have these mucous membranes, these wet epithelium that are covering up my nerve ends, which are going to absorb those smells, and they're going to go in. They're going to open my gates and send an action potential. So that's kind of where we're headed with this. So what are we going to see? We're going to see dissolved chemicals, which are going to open my gate and send my action potential. Where is it sending it? It'll send it up the nerve endings to the olfactory bulb back to the brain. So does that help? And we're going to walk through it. So you just jumped a little bit ahead of me here. All right, but basically what I wanted you to remember is we can open these gates to send an action potential. How do we send these back on these nerves? This is cranial nerve number 1, right? How do we send a signal down our nerve? We do it using action potentials. How are we triggering an action potential for our sense of smell? We're opening chemical gates. All right, so we are going to have basal epithelial cells. Do you think that your epithelium in your nose is subject to a lot of wear and tear? Yeah, it is. Why? Because it has direct contact with the outside world, right? We're breathing in dust, we're breathing in dirt, you squeeze your nose to blow your nose. We do all kinds of stuff to our nose, which is hard on our epithelial tissues. So our basement, our lamina propria, is going to have stem cells in it. What are those stem cells going to be doing? Reproducing cells, mitosis. They're my mitotic cells. And our underlying lamina propria, what did we say lamina propria was? Yeah, which is the connection to my connective tissue. Why do they have to sit on a connective tissue? Because they're avascular. So what is my connective tissue giving them? All the stuff that blood would give them, right? Nutrients, oxygen, plus my epithelial tissues are giving the connective tissue their waste, their water, things like that. So my connective tissue almost acts as my blood supply. It doesn't almost, it does act as my blood supply for my epithelial tissue. So we have to have the epithelial, we have to have a connective tissue, and we're going to have our nerve endings in there. So when we're talking about our lamina propria, we're talking about, you know, that connective epithelial combination there. What is the tissue that most of our epithelial tissue sits on as a connective tissue? Do you remember from 201? What type of connective tissue? No, it's airy. Yes, loose areolar. This is the #1 connective tissue that your epithelial tissue sits on. Why? Because it's airy, so stuff can diffuse through it easily. And that's what we see here. Our connective tissue for our olfactory epithelium is areolar tissue. And we already said it's going to be a mucus gland. Why would we want a mucus gland? I already named one reason why. Yeah, it can protect, so your mucus is going to prevent a lot of bacteria and things from joining in there. It's going to trap things, but what else? How am I sending an action potential? What am I opening to send my action potential? What channel? Through a chemical channel. So somehow I have to get chemicals to here. What's my epithelium going to help do? Or my mucus, I mean? Trap and absorb those chemicals from what we smell. So your mucous membranes help to grab onto and absorb those chemicals so they get to these gates in my nerves. All right, everybody with me so far? All right, so this shows the nose right here. Here's some of the anatomy. You guys learned a little bit of this last time. Here's our olfactory epithelium. They're just showing this with kind of this purpley stuff here. Here's my olfactory tract. We learned about that already, right? Yeah, everybody good with that? And our olfactory bulb. These are our free nerve endings. What on a nerve cell, what end is the receiving end on a nerve cell? Let's do the anatomy. Well, on the nerve cell, I'm going to draw a silly little nerve cell. What'd you say? Yes, dendrites. These are my receiving end. These send my signal away, right? And this is my cell body, my stoma. So what's going to be hanging down in my nose? What's going to be my receiving end? Dendrites, right? So these guys right here are dendrites to my neural cells hanging down for my sense of smell. Well, I have my epithelial tissue, which is going to secrete mucus for protection, right? We have to be sitting on a connective tissue. What's our connective tissue going to be? Loose areolar connective tissue. What's the function of that connective tissue? to support the epithelium, right? It supplies blood vessels, all the stuff epithelial tissues need. All right, so here comes our odorant. Luckily, this is a very lovely smelling odorant. It's going to come up into the nasal area. You guys might remember the conche from 201. But these fumes, these odor fumes, are going to come attached to the mucous membranes of the epithelium. And guess what they're going to do? They're going to attach to chemically gated channels in my nerve endings on the dendrite side. So that's what we see right here. Which nerve does the sense of smell? Cranial nerve 1, olfactory nerve. All right, so this is just a big giant zoom in. Here is our cribriform plate that had our cribriform foramen. So this is our ethmoid bone. You guys remember this? And we had all those tiny little holes beside the crista galli that were the holes. And this is where my nerves go right down into the top of my nose, is through all those little olfactory foramen. Here's my lamina propria. This is my connective tissue, my area where my connective tissue adjoins my epithelial tissues. And here's my epithelial tissues. What are they doing? They're protecting my nerve endings. They're secreting mucus, which is going to help with absorption of chemicals, trapping particles, things like that. All right, so here are my free nerve endings here hanging down in this epithelial area. Here comes my substance being smelled. It's going to get absorbed into my epithelial tissues and it's going to attach. Basically, it's going to end up attaching to our voltage gate. If you see here, these are my dendrite. And this is kind of my mucousy membrane right here. And you can see these epithelia are supporting the structure of these dendrites as well. All right, so this is going to create a potential. I don't care if you know if it's G protein coupled or not, but it's going to create a potential or an action potential or a depolarization. What did that mean? That meant I opened up my chemically gated ion channel. Sodium began to flow inside the cell. What happens to my voltage when sodium flows in? It was at a -70 difference. What's it going to do? It's going to go closer to 0, which causes these voltage gates to open, right? Potassium's going to go out because of the concentration gradient. Changes my voltage out here. Voltage gates open, voltage gates opens. We just begin this ripple cascade effect of all these gates opening and closing, which ripples my signal down my neuron. All right, on our olfactory pathways, our afferent fibers leave our olfactory epithelium. What does afferent mean? Yeah, the sensory, and they're headed into the brain, right? And we just talked about how do they do this? They do this depolarization. They're going to travel up through the cranial foramina, and they're going to hit the olfactory bulbs. And so they go up through the cribriform plate. They reach the olfactory bulbs of the cerebrum. And do you remember where those olfactory bulbs were sitting right out on the bottom of the cerebrum? And this is our first synapse point. So these are our sensory nerves that were hanging down. They receive the signal. They send it up and through the cranial bone of our ethmoid. Synapse is on the olfactory bulbs. So now the axons that leave the olfactory bulb at the synapse point are going to travel along to the olfactory cortex, to the hypothalamus, and to the limbic system. These guys also help to process memory, right? Our limbic system. There is a lot of memory tied to smell, right? You walk in, I know when I was a kid, we'd walk into my grandma's house and it had such a distinct smell. If I ever smell that smell, I instantly think of my grandmother and memories about my grandmother. So there's a lot of memory tied to your sense of smell. And you can see because some of this runs through these same areas of the brain. The olfactory information is the only information that goes directly to the cerebral cortex. The rest of them are going to synapse or not get routed through a nuclei. What was the nuclei in the brain? Remember, we had like basal nuclei. They're groups of cell bodies where then we routed stuff. With the olfactory, because it's right under the brain, most of our nuclei were found in our brainstem, right? And so it's not going to take it all the way to the brainstem, sort it, and then send it back to the cerebral cortex. So what it does is it comes in and it goes directly to the olfactory portion of the cortex to where we can get our information. The last sorting point, remember, of all your sensory stuff was the thalamus. The difference here is that the olfactory information does not go through those. All right, and I don't ask you to know all the voltage changes and things for that. What I will mention, because we're going to talk about gustation, which is your sense of taste, is taste has to do with chemicals, right? And if you're tasting stuff, those specific things oftentimes are also secreting an odorant. So there is the connection of absorbing an odorant as well as tasting a taste, which ties those two together. So sometimes people will say, well, if you don't like the taste of it, like you could plug your nose and eat it, right? And you won't taste it. You have a stuffed up nose because you have a cold, you don't taste your food as much. Because those chemicals are very tied together, kind of in how our brain perceive those because of those chemicals. So it is true that your sense of smell plays a big part of your sense of taste as well. All right, I don't ask you to know any of this stuff, so don't worry about that. All right, olfactory discrimination. This is kind of interesting. We have thousands of different chemicals that we can pick up the sense of, but dogs have 72 times more olfactory receptors than us, which is why dogs can smell stuff out so well, and they use them for such specialty things. Like you have your cadaver dogs and your drug sniffing dogs, and you have your therapy dogs that can detect blood sugar levels. How do they do it? Sense of smell. They can smell sugar, they can smell illness on people, things like that. Why? Because they have a very, very heightened sense of smell. They have a 10,000 times better smell than we do. Olfactory receptors are one of the nerves that regenerate really, really quickly, really, really easily. Why do you think that is? Think about your receptors of your sense of smell nerves. Where are they hanging? Right into the top of your nose, right? So they have direct contact with the outside world. Are they going to sustain a lot of damage and a lot of wear and tear? Yeah, absolutely. So this is one of the nerves that really regenerates readily, easily, quickly. Why? Because it withstands a lot of wear and tear. As you age, though, it is true that as you age, your sense of smell kind of declines. Why? Because our neurons decline with age. Why? Some of it's hormonal. As we talked about before, as you get older, we just don't regenerate tissues as easily. And the same is true here. So as you get older, you don't regenerate those receptors at the same rate, so you don't smell as well. All right, gustation. So that's smell. Not too tricky, right? So it comes into our nose. It routes up to the through the cranial foramen, and what's the first synapse point? Olfactory bulb, and then those go back to the olfactory tract to where? To the cerebral cortex. They go directly to the cerebral cortex, where now our brain will be like, hey, I smelled this smell. It's kind of a learned thing though, right? Babies probably probably don't have the same number of sense of smell. They have receptors, but you kind of have to learn what those senses of smells are. They do know some smells though, right? For sure. Yeah, they definitely know like the learned smells. Yeah, they know mom smell, they know dad smell. I saw a cute little, I think it was a TikTok video or something. And it was a dad holding his baby, and it was like, my baby only loves my wife. And he had like this fake face of his wife right here, and he's holding the baby. I was like, I'm pretty sure that baby knows it's not his mother or bud. All right, gustation. This is your sense of taste. This is going to give us obviously lots of information about chemicals that we bring into our mouth, foods, liquids, things like that. These are also going to be epithelial. Are we going to have a lot of wear and tear in our mouth? Yeah, obviously, right? You burn your mouth and it hurts your taste buds. So we have to be able to regenerate. Remember, one of the characteristics of epithelial tissues is they're easily regenerated because they withstand a lot of wear and tear. So we are going to see that these guys can regenerate pretty easily. What we basically have on our tongue is we have different papillae. And if you remember from when we talked about the skin, we talked about the dermal papillae, the little upheavals. Do you guys remember that? Papillae just means a mound. So we have all these little different taste receptors on our tongue and that are different shapes, but they're basically these little mounds that stick up on your tongue. Different mounds have different papillae have different number of neural receptors. They detect different tastes, different information about the chemicals. How do you think we send the action potential with taste receptors? How would we open our gates with a taste receptor? Chemical, voltage, or distortion? It's going to be chemical for sure. All right, so our taste buds have these receptors. They are distributed. You should probably know this. I think I might have a test question about this. Where are they distributed? They are on the tongue, the pharynx, and the larynx. What is the pharynx? I feel like nobody ever knows what the pharynx is. The pharynx is shared tubing between digestive system and respiratory system. So anytime these are sharing tubes, we call it the pharynx. At the point it splits into just digestion and just respiration, we change its name. Who's the little trapdoor that makes that split? The epiglottis. So at my epiglottis level, I'm going to split and go back into my esophagus, which is going to be my digestive tube, or go forward into my trachea, which is my respiratory tube. So prior to that split point, this is going to be my pharynx. What's my larynx? It's your voice box. So a little bit of the top of that, right where your epiglottis is, right at that same location, you're going to have some of your taste receptors. So all the way down to here, you have taste receptors.
Your tongue's pretty long.
Yeah, it's weird because it doesn't just blunt end at the back of your mouth. It kind of goes down the pharynx area just a little bit. We have lingual papillae, and I said those are little mounds. We'll talk about the various types of mounds, but we have some papillae that don't have taste receptors. And these guys are just helping to move food around. When we chew, when we begin to digest, we mechanically break down food. We also chemically break down food. And we have some of those little projections on our tongue that just help to move the food around and put it where our teeth can find it and grind it up. So not all of your taste buds, well, your taste buds do, but not all of the papillae in your tongue are taste receptors. Okay, these are the ones that you have that are taste, well, these aren't all taste receptors either, but we have our filiform papillae. These guys don't have taste receptors. These guys just move your food around. These are the ones that move your food. They're kind of sharp and pointy papillae, kind of like this. They have sort of a rough surface to them. I guess they're a little more triangular, actually. I don't know if I can draw it very well. I have a picture of it. But these are the friction creating ones that help to, along with your tongue, to manipulate and move your food around for chewing and tasting and things like that. As we chew and we crunch, we're kind of releasing those chemicals that then can go to these other papillae, which actually have our sensory receptors in them. One that is a taste bud is the fungiform. This looks like fungi, right? Which is a mushroom. These are mushroom topped. They look like the top of a mushroom. You should know that there's only about 5 taste buds in each of these. So not very many taste buds in these kind of mushroom topped ones. We have our valet papillae. These are really large and they kind of have layers to them. You'll see a picture of them here in a minute. These ones have about 100 taste buds. And then your foliate ones also have taste buds. They're not worthy enough to give a number to, evidently, so. Everybody good? Okay, we are going to have still an epithelial tissue, which we know this, right? Because it has direct contact with the outside world. So we're going to have gustatory or gustatory epithelial cells. And basically what we're going to see is we're going to absorb those chemicals, just like we kind of saw with the nose. Those epithelia are going to absorb those chemicals from our food, and they're going to be kind of grabbed by these taste hairs and absorbed through these taste hairs through a taste pore. So that's how we enter into where the nerve is. The nerve isn't directly on top of our tongue like we saw for the nose. That would not work out very well. So what we do is these little villi kind of grab that chemical. What do villi and microvilli do? Increase surface area for absorption, right? So it's going to absorb that chemical down kind of into the inner parts of the taste buds through the taste pore, which is where we're going to see our action potential triggered. And we said it's going to be a chemically gated channel, right? That's going to open up and send that action potential. These little microvilli or taste hairs survive for about 10 days. So they're rapidly overturned. Why? Because they're sustaining a lot of wear and tear because they are out in the open area of the tongue. These guys, your gustatory nerves are innervated by cranial nerves that go to the medulla. At that point, it's going to travel to the thalamus. And it's going to go to the gustatory area of the brain, which is the insula. Where do we find the insula? It was on the, lateral sulcus. It was a little island we said down in the lateral sulcus. So that's kind of where your taste information goes. But it comes in, it goes to the medulla, routes up to the thalamus, and then goes to the insula. This shows our, was everybody done? Did I go too fast? Are you guys okay? All right. This shows our tongue right here, and I'm going to talk a little bit about where you have your most abundant taste. We kind of have sweet and salty receptors. We have a more abundance of those kind of out on the anterior portion of our tongue. We have sort of our bitter sour taste back here in the back. We have a sensation called umami. Umami kind of gives us our savory sensations. And here's a picture of each of these. Here's our valet. Remember how I said it was really big and it had kind of rings to it? These are all taste buds. And down in here, this is the one that we said had the most, right? The most abundant. And those chemicals come down. They're going to go to these receptors in these taste pores. And then we're going to see an action potential fired through chemically gated channels. And it goes back. Where does it go? First in the brain. Before that, to the medulla. It's a sensory information from the medulla. Where's it go? Thalamus to the insula. You got it. Here's our folate papillae. These guys don't have very many. We said about 5-ish. Again, the chemicals are going to come down into these pores, get absorbed through, and fire off our action potential. Here's our fungiform. It looks kind of like a mushroom top. And then here's the one, these are the guys that just do the movement, these filiform. They're what make your tongue so rough. They really make like a cat's tongue rough, right? You do. You can actually damage the epithelium. You probably don't burn your taste buds, but you burn this epithelium up here, and we have to regenerate it. So. I don't think so. I have never heard that. So
Oh.
have you guys heard that before?
I don't know, it's something that they didn't use, like straight sugar. Then they put that-
I've never heard that.
Yeah, like a beef burger.
Yeah, I've never heard that either, so I don't know. Okay, so here's kind of a drawing of our what we're seeing. And let me backtrack and kind of show you where we're looking. When our chemical comes down in here, we're kind of looking at this junction right here at this taste pore where that chemical's going in. So this is a blow up of that. So here would come our chemical. Here's our little microvilli. Microvilli increase surface area for absorption of things. So here comes our chemical in. It's going to open my chemically gated ion channels, and boom, it's going to begin to send my signal. These guys use a transitional cell, and they are supported by our epithelia, our gustatory epithelial cells. That kind of helps hold those into a format. And this transitional cell sends it to these neurons, which then send it back to the medulla.
You can change shape.
What do you mean? What change shape? No, they don't. They just send the chemical just passes through it. Is that what you mean? Or do you mean it has some flex to it?
I was kind of thinking, like the uterus. Or the,
Oh, no.
transitional cell.
Oh, no. I see what you're saying. No, it doesn't. It's not the same. No, not the same at all.
Okay.
All right, here's your four primary taste sensations. You absolutely should know this, and you should kind of know where they're most abundant. You have all of these all over your tongue, but where do we have the most amount of sweet receptors? Out on the anterior surface of the tongue. Where are the most salt? Anterior surface of the tongue. Sour and bitter are posterior tongue areas. You're much more sensitive to sort of your bitter, sour, unpleasant taste than we are to our sweet and desirable taste. So our gustatory receptors tend to be much more sensitive with those bitter and kind of undesirable tastes. Probably because stuff that doesn't taste desirable oftentimes isn't good for us, right? Like if you were to taste a poison or something that you thought might be okay to eat, yeah, and your taste receptors are really sensitive, it's almost like a warning mechanism of, hey, that's really bad. You probably shouldn't be putting that in your mouth. So that's why I think we tend to have those more sensitive to bad stuff. Umami, as I talked about, it's kind of your savory receptors. We find these in our valate papillae. Those were the big layered ones that have the most amount in them. And they actually secrete, when you get those really savory tastes, those, and they trigger those umami, it actually causes a chemical glutamate to be secreted, which is what gives you that sensation of that really savory taste. So what are some ideas or some types of food, maybe broths, things like that?
Did you say broths?
Broths, like a soup broth and gravies, things like that. Well, anything you kind of classify as savory, that's kind of what triggers that. We have water receptors. The bulk of your water receptors tend to be found in your pharynx. And I don't know if you've ever had that sensation where you're really, really thirsty, really dehydrated. Maybe you ran and you're dying of thirst and you drink and you get that really satisfied feeling back in your pharynx and kind of your deep throat area. That's because that's where you have those big water detectors back there. They say water doesn't have a taste if it doesn't have chemicals added to it, but you still get that real sensation of contentment because of those receptors back there. Taste sensitivity varies from person to person, and that is a genetic component. So genetically, you're going to be predisposed to maybe higher sensitivity or more taste buds to certain types of flavors. Children have a really sensitive sense of taste. And as you get older, you tend to lose some of your taste buds and you don't regenerate to the point. So children are really sensitive. Like think about spicy things. As we get older, we like spicy stuff, or we tend to at least like it more than we did when we were younger. Young kids are really sensitive to like spicy things like that. That's because they do have more taste buds, more receptors, and they're very sensitive to it. But there's a big hereditary component to your taste buds and how sensitive they are and what flavors they're sensitive to. One genetic thing is the PTC. Do you guys ever done the little genetic test in like Bio 111? I don't think we did it here because we just didn't have enough time. But you do things like, you do the dominant and recessive traits like hitchhiker's thumb and can you roll your tongue and all those different things. One of them is the PTC. This is a simple dominant recessive trait, whether you can taste, they put it on a piece of paper and you taste the paper. Some people can taste it and it's really bitter, sour, horrible taste. Some people can't taste it at all, just as like licking a piece of paper. Yeah, so I don't know if we have any. If we do, maybe next time I'll try to bring it out and you guys can test yourself and see. We might have some back there. I'll look and see.
So if it's like DNA, is it like when you are pregnant, what you're or like?
No, it's more genetically predetermined. Like with this is the recessive, dominant recessive trait. So you either inherit the taste receptors for PTC or you don't. And if you have them, you taste it. And if you don't taste it.
So if you have a gluten thing and then they inherit it.
That's not anything gluten, like a gluten intolerance.
Yeah.
That has nothing to do with your gustatory receptors. Yeah. Gluten intolerance is more of a digestive system enzyme. But some of those, like lactose, is a genetically inherited thing if you're lactose intolerant. And basically, you aren't producing the enzymes that break lactose down. You don't have the lactase enzyme. I think you might be mixing those two up. Are you thinking along those lines? Yeah, you're jumping into some of the digestive system stuff. Right now, we're just talking taste. Yes. Yes. With age, I don't know about that on the lactose intolerance. Really interesting. I don't know the answer to that, but that's a good question. Yeah, so if you're online, she asked if there was an age factor to the ability to digest lactose or not. And that I don't know. I don't think so. I think you either produce the lactase enzyme or you don't. Oops.
Yeah, I think I'm super good.
Oh, and this is supposed to say we are much more sensitive to unpleasant taste than pleasant taste. I already kind of mentioned that. I don't know why it got cut off here. And it's sort of a protective mechanism. Why? Because really unpleasant tasting stuff, some of your poisons, things like that, is kind of a warning system. Does that mean every unpleasant thing is bad for you? No, sometimes we just don't like it and we're more sensitive to it. Everybody. Oh, I like Brussels sprouts.
Not the right thing, but just like plain in the microwave bag.
All right, I think I will go just a little bit farther. I'll train you guys loose early. I'm just going to do the general anatomy of the eye, and then we'll stop before we get into the function of the eye. And you guys are going to be like, I'm a rock star at this because we already did a lot of this in lab. So on our eye, our vision is going to be one of the things that obviously we're going to be looking at in the eye. We have a lot of accessory stuff to our eye that helps to take care of the eye so we can see. What are some of our accessory structures? These are going to be eyelids, superficial epithelium. We've seen epithelium in everything we've talked about so far. This part of the sensory organs, right? We saw epithelium in our nose. We saw epithelium in our taste receptors. We have epithelium covering the surface of our eye. If you remember back to 201, we said anything that has direct contact with the outside world is going to have an epithelium on it. All right, so we also have lacrimal apparatus. Anytime you hear the term lacrimal, it's referring to tear ducts. So lacrimal means tear ducts or portions of the tear duct. Our accessory structures are going to provide protection, like our epithelium of the eye is going to be providing protection. It does lubrication. Lubrication is also a protective function, right? If we keep the eye moist, we can wipe away bacteria. We have enzymes in our lubrication, which can attack any foreign bacteria pathogens that come into our eye. So I feel like lubrication kind of goes with protection as well, but it also is protecting from friction and abrasion as well. All right, everybody good? All right, eyelids. The proper name for an eyelid is called the palpebra. And it is a continuation of the skin. So your eyelid is actually some of that epithelial, that epidermis of the skin. When we blink our eyelids, we are going to lubricate and basically wipe away the surface of the eye. Which means we're going to have to look at some point down the road here on our anatomy of how we get the lubrication into our eye. Right? So we're going to secrete lubrication. Our eyelid is constantly blinking that lubrication across our eye. It has enzymes in it which can attack bacteria and pathogens, but it's also going to be reducing friction. Our palpebral fissure, what's our palpebra? Eyelid, so the fissure is the slit in our eyelid, right? So it is the gap that separates basically our upper and our lower eyelid is the fissure. And then our eyelids have a medial and lateral angle. You also hear it called the canthus. So out here on the sides where your eyelids come together, this would be my lateral. Where they come together in the middle is my medial. I'll show you a picture here in a second. Yeah, so lateral, medial. Eyelashes. Without looking, what's the function of our eyelashes? Yeah, keep debris away. And if something brushes our eyelashes, what do? We slam our eyes shut, right? So it protects our eyeballs from objects coming at our eye as well, but it also acts like a little umbrella and keeps stuff from falling in our eyes and things like that. Tarsal gland. These are the glands that actually secrete part of the lubricants in the eye. So they secrete a lipid-rich product, and this helps keep our eyelids from sticking together. So when we blink, we don't have to like overcome the adhesion of our two eyelids together. This helps to make it really easy to push them together, pull them apart. Lacrimal, what is that going to be talking about? Our tear ducts, our caruncle. This is the soft tissue at the medial angle of the eye that has the glands. And we'll look at a picture. But you know, when you look at the corner of someone's eyes, you can see a little tissue in the corner. That's what we're talking about. Is everyone good? Anybody want another minute? All right. So here we're looking at our eye. This is going to be our palpebra. This is the fissure, the opening between our upper eyelid and our lower. Here's our medial angle. So it's where our two eyelids come together. This is going to be my lateral angle over here. Here's our eyelashes, obviously. Here's our caruncle. It is that little tissue in the corner. This is where we see some of that secretion area from. Yeah. It does get kind of itchy, and because it's a lipid heavy protein, but we also get debris and we're constantly swiping that debris away. That's why sometimes like at night and then you wake up and you have the sand from the sand man, it's kind of residuals from all of that stuff. All right, conjunctiva. This is actually the mucous membrane that's covered by an epithelium. So our palpebra conjunctiva covers the inner surface of the eyelids. So it's on the inside of our eyelids. We have the bulbar conjunctiva, covers the anterior surface of the eye. And then what do we have when we hear conjunctivitis? What does itis mean? Yeah, inflammation of. So this is when you get a really red surface of those membranes to your eye. That's what we'll call conjunctivitis. Pink eye is a good example of conjunctivitis if you've ever seen anybody with pink eye. But not all bright red eyes are pink eye. Pink eye tends to come from, I think it's a bacterial infection specifically. No, this is a pink eye. Is that what you mean? Yeah, but pink eye specifically is a bacterial transmission that causes that redness. You could get red eye or conjunctivitis from lots of different stuff. So this is not just pink eye as far as the bacterial pink eye, but we can get a pink eye from other things. Lacrimal apparatus, we said this is going to be kind of our tear ducting stuff. This is going to produce, distribute, and remove tears. What is our lacrimal gland? This is going to be our tear gland. And it produces tears and it goes over that conjunctival surface, so over that mucous membrane surface on our eye. And remember how I told you it contains enzymes, which are going to take care of pathogens and things like that? Those are lysozymes. So these are antibacterial enzymes that are in your tear duct fluid. They do a pretty darn good job of keeping stuff from entering our body through the surface of our eye. Can we still get stuff? Yeah, it's a mucous membrane. And because it's a mucous membrane, it can be absorbed into the inner parts of the body through the lacrimal ducts, things like that. But these guys do a pretty good job of keeping it at bay. All right, tears collect in a lacrimal lake at the medial angle of the eye. So when you look at somebody really closely at the corner of their eye, it can sometimes look like they have a little pool of water. It's not always very obvious. And then we have a duct called the nasolacrimal duct. So your tear ducts actually go into a duct, a little hole. Through the bone part of it is called the lacrimal canal, but it takes that tear that came from your eye, it washed across your eye, and then it drains at the medial angle of your eye, and it goes back through the nose area. And yeah, that when you're like, oh, that postnasal drip kind of, it's kind of that same area. All right, so this shows some of this. Here is our lacrimal gland. So your tear duct secreting gland is kind of up on the upper outer part of your eye. It's going to secrete tears. You blink it across your eye, and then it drains on the medial lower part of your eye. So it kind of goes from here across to here. Here's our gland. This is going through the lacrimal canal. If you guys remember, we talked about that in 201. So we're just kind of flushing tears across here, draining them here. So I want to tell you this cool story. You guys want to hear a story? So my dad is a veterinarian, and when I was a kid, he treated horses a lot. He's retired now, but we had a horse that just kept getting these horrible eye infections all the time. And we had these big hay feeders because you have to feed them off the ground because they'll pick dirt and stuff up off the ground. And if they get sand and dirt in their belly, that can kill them for other reasons. So we always have feeders, right, that are up off the ground. And she would jam her head into the middle of this feeder all the time. And we had alfalfa with little flakes and stuff. But she kept getting these horrible eye infections. And so he came out, he kept treating them superficially. And finally, he's like, I'm going to flush her lacrimal gland. So he takes this tiny little tube and he sticks it into her nose where the drain for the lacrimal duct comes into your nose area. And he sticks that tube right up in the inner part of the nose into that gland and just shoots saline. And it all comes out her eye. And it's all just huge glumps of alfalfa and dirt and all this stuff. It was very interesting though, because I mean, I couldn't have even thought she could have that much stuff. But her entire tract here was just stuffed full of dirt, debris, alfalfa, and that's how he flushed it. It was super interesting.
So why if you blow your nose or like air?
Yeah, you'll get that little air section up to your eyes. It's because you're affecting, you're basically, when you pinch and blow, you're blowing air back up your lacrimal duct. Yes. Oh, sorry. The lacrimal, the nasolacrimal canal. I think that's the one you want. You want me to backtrack? Oh, the cannuliculi. The canal, it's the lacrimal canal or nasolacrimal duct. Okay. And this is what he flushed right here with the nasolacrimal duct. I thought it was really cool, actually. Yeah, there's a little drain that comes kind of into the top. It's pretty high up. And you have a little opening and it'll just flush. That's too why when you cry, what happens when you cry? Your nose starts running, right? It's because you're shedding more tears and they're draining down into your nasal area. All right, eyeball. We have an outer fibrous layer. A lot of this stuff, I think you guys are going to feel like, hey, I know this stuff. You have a vascular layer and you have your deep inner layer, which is your neural receptor layer, which is the retina. And we learned about that. What do we call our outer fibrous layer? Do you remember? The sclera, yes. And then we had our uvea. We didn't really talk about that but very much, but we will. And then the retina was our receptor layer, right? That's where we had our neural receptors. We have a lot of fat around our eye. We saw that on our cow eyes, right? We cut those big, enormous globs of fat out on some of them just to even expose the iris of the eye. Why? It's to protect our eye from blows and things like that. Your eyeball's hollow. It's filled with fluid. We already learned 2 of the types of fluid. What are they? Aqueous humor and vitreous humor. Aqueous humor circulates from the anterior portion of the eye into the posterior portion of the eye. So it kind of goes back and forth. It's kind of like your circulatory fluid of the inner eye. And where did we find the vitreous humor? Anterior, posterior portion. Posterior portion. And what was its function? Yeah, it holds the retina kind of up. So it protects kind of the shape and the structure, the flexible structure to the back portion of the eye. So it gives support with that sort of thick shape to it. All right, our anterior cavity we saw was very, very small, right? How do we kind of define the anterior portion of the eye? Lens and forward, right? So everything out just in front of the lens. Once we hit the lens and we went backwards, that was our posterior portion. And our posterior portion of our eye, much larger than our anterior portion of the eye. And you can see our aqueous humor is anterior. It actually goes into the posterior section, and our vitreous is in the posterior. So this shows kind of our lens right here. This is our anterior up here. This is going to be our posterior here. Here's our ciliary bodies. We saw those. It's kind of hard to tell from this picture, but our ciliary bodies. When we look at the outside, this is our sclera. Remember, our sclera became our cornea across the front. It was just the thick white part on the back. And then this inner lining right here was our neural layer. What did we call our neural layer? That was our receptive layer, the retina. And what was the layer between those two? The choroid, and it basically reflects the light. So the eye is super interesting. We've seen that we've received things so far and they've gone directly down the nerves. What the eye does is light comes in and it has to bend. We call our bend refraction to our light. The number 1 refractor to our eye, remember we're shooting for that focal point in the eye. What do we call our focal point? The fovea centralis, right? So we're going to try to bend that light to hit where our fovea centralis is. Our cornea is our number 1 light bender. It does the most amount of light refraction, is up here on the cornea. What is astigmatism? Anybody know?
The cones are things it's not sort of shapeless.
You're exactly right. It's the cornea though. So if I was looking at the front of my eye, I'm gonna turn my eyeball sideways to you. So this is the front of my eye. Your cornea should look like this. When you have astigmatism, it does this. It kind of gets elongated, almost football-y shape. Who did we say is the number one light bender? The cornea. So if I misshape my cornea, am I going to refract my light as easily to my fovea as I want? No, which is why when you have astigmatism, sometimes that makes you blinder than if you were nearsighted or farsighted, right? And that's because it bends your light. So this cornea and the shape of it is super important. Why? Because it is refracting, it's bending the light towards the fovea centralis. And what is unique, I started to say this, is when we look at the eyeball here, the light comes in the cornea. I'm just going to draw a straight line, even though here would be our cranial nerve. What is our optic nerve? 2. This is CN2. What's it do? Is it sensory or motor? Sensory. So it does my sense of vision. So I have my outer layer. What do we say this really thick fibrous outer layer is? The sclera, and then a retina, and here's my neural layer. And I have my choroid in between, right? Your choroid actually acts, they call it the pigmented layer, and you guys saw it was kind of black. In the cow, it was unique because it had some of that tapetum to it. But basically, light comes in, it kind of bounces off the choroid layer and gets received from the retina going back towards the anterior portion of the eye. So the light doesn't come in and get received from the retina here. It comes in and it goes to the choroid, and then it gets received as it goes back out towards the anterior portion of the eye, which is kind of unique about the eye. All right, so we talked about this. This shows a side view. Here'd be my cornea. Here's my anterior portion of my lens. This is all going to be posterior. Here's my sclera, my choroid, and here's my retina. Here's my fovea centralis. This is my focal point. Oris aurata, what did we say the oris aurata was? Yeah, it's the lace-in point where our retina joins in there. All right. Our vascular layer is the uvea. And this gives the blood vessels. Obviously, it's the vascular layer, right? It's going to do blood vessels and lymphatics. We always have lymph vessels. Wherever we have blood vessels, we have lymph vessels. This area helps to regulate the amount of eye coming into the eye. It secretes and reabsorbs our aqueous humor. And the aqueous humor, remember I said it's kind of like a circulatory fluid for the eye. So it's circulating through anterior and posterior portions of the eye. And this also is going to help control the shape of the lens. As we fine tune the shape of the lens, that is the second thing that bends light. So our cornea, we have no control over, right? Our cornea is what it is. We hope it's the right shape. When we get into our lens here, these are attached to little ligaments, and we can either make our lens fat and round, or we can elongate it and make it skinny. Depending on what we do, we bend the light again, which can change this focal point at the fovea centralis. So that's how we can adjust. We can look out far and then we can read up close. Those are going to take different focal points by bending my light to my fovea centralis, but I can do it by squishing and elongating my lens to my eye. Is everybody good? Yes.
I had scratches on my cornea. Actually, I had to go to the. I couldn't.
Oh, geez.
So whenever I would open it, my eyes would just water and it would hurt so bad, but I could barely see. I think they said like,
Wow.
why is that?
That affects your vision.
Yeah, whenever I just like
Well, your cornea does refraction maybe with the injury right there. I'm guessing. I don't honestly know, but I'm guessing maybe with the injury of the tissues, it's changing your light refraction. Or maybe because of the sensitivity, your lens isn't adjusting. I don't know. I'd be curious.
Not even just my hand, but I put a towel there and then my hand, like hurt
Ugh.
my, it feels bad.
Yeah, eyes are one of the fastest healing tissues in the body. We had a friend that last 4th of July, they were having a 4th of July party. No, we weren't supposed to, right? I wasn't at it. I just heard about it later, but they were shooting off bottle rockets and stuff like that. And they had a rogue bottle rocket. She was just sitting in a chair. She wasn't even close to it. She had her son on her lap and it came and it hit her right smack in the eye. Like, what are the odds, right? And it goes and it hits her right in the eye and it actually detached her retina. So her whole retina came off the back of her eye from the impact of it. And it actually detached her iris. So her color, it looked so weird. Remember how I said the iris is the color of your eye? When you looked at her, instead of having like, here's your pupil and here's the color of your eye, it was all collapsed in like this. It looked really weird like that, and it detached part of her lens. So they had to let her eye heal a little bit and the trauma to go down in her eye. And then she had multiple surgeries, and they went in and reattached her iris and they repaired the ligaments to her lens. But her retina, they did some reattachment on her retina, but she actually damaged her macula too, the way it impacted into her eye. And I saw her recently at graduation and I was asking her, like, how's your vision now? And she goes, it is so weird because at first I couldn't see anything out of it at all. And she's like, if I cover up that eye, I can't see anything. It's like prisms and light smears and everything. But now when they're both together, she's like, I see perfect. I can't even tell that I can't see out of that eye until I cover it up. Yeah, your brain compensates. There's a study that they did where they took people because your brain really is a big part of your visual processing, obviously. But you can kind of trick your brain into learning stuff about your vision. And they did this study where they gave people, so when you see stuff, you actually receive it upside down and reversed image into your eye, and your brain turns it around so it appears right side up to us. So they did this study and they gave all these people these glasses that flipped everything upside down. So when they were walking, like it seemed like they were walking and everything was upside down and everything, they had to wear these glasses. I can't remember how long it was. I want to say it was like 3 to 5 days. And their brain turned everything right side up. So as they're walking with these glasses now, the whole world's right side up. Then they took their glasses off and everything was upside down. So then they had to readjust to normal without the glasses, but their brain flipped it around. So your brain can really compensate for your visual processing. It takes a little bit of time, but that's what it's done for her. And she's like, when both my eyes are open, I can't even tell. But if I cover up my good eye, I can't see anything. She's like, I'm legally blind in it. Yeah, it's mostly your detached retina and her damaged macula. She hit her macula, which is your cones. So she kind of destroyed almost all of her cone reception. So she sees no color. She said it's just like big blobby streaks of light. That's what she sees out of that one eye when it's just that eye. So, but it was the craziest thing looking at her iris till she got it fixed. Oh, oh, interesting. Well, from the lens, it was the lens. Oh, interesting. Yeah, it's just a protein, and maybe if it wasn't firm enough, maybe with the ligaments, the way the ligaments attach into it, it wasn't holding it. Well, it would all be about refraction. It would go back. So if you had a fold, you might have 2 refraction points, maybe, I'm guessing. Because your lens is your second biggest light bender. So maybe with that fold, she was getting multi-refraction. I don't really know. I'm speculating, but that's interesting. The really cool thing is now they can go. So when you get cataracts, it's when we were looking at the cow and you saw how we denatured those proteins or and they weren't clear. That's basically what a cataract is. Our proteins begin to get cloudy. We can't see very well. Well, now they can go in and actually give you a fake lens. They can totally replace your lenses. My sister had it done. She's also had a detached retina, but she was so blind. She had like Coke bottle glasses, like the worst ones you've ever seen. They were so thick. So she wore contacts all the time and she just kept getting worse and worse. They couldn't get her vision corrected. So they actually went in and did a double lens replacement. And she has one lens that she didn't do them equal, so she doesn't need reading glasses either. One eye kind of sees, and again, your brain kind of compensates for that. So she has a reading lens and a distance lens, and she can see perfect. Kind of crazy. Okay, so this just shows this. When you get the picture of the back of your eye, you can kind of see. Have you guys ever done that? That Optomap at the eye doctor, and they shoot. Have you guys done that? And they shoot the picture. Oh, it's really cool. Or they dilate your eyes and then they look into the back of your eye. You guys have never had that either? Yeah. Well, they have new machines called Optomaps, and you sit there with your chin and they like flash a light in, and basically they take a picture of what they would do when they dilate your eyes. So now you don't have to have your eyes dilated. I think they charge like 30 extra bucks or something for it, but it's totally worth it. 25. Yeah. Oh, so much easier because you don't have to get your eyes dilated. No, it doesn't hurt at all. It's just like a flash of light in your, they have these little, this greenhouse, and you sit there and have to track the greenhouse and it takes an image of your eye. But anyway, when they look at those, then it's really cool because they pull it up and you can see the picture of the back of your eye. But they basically look at your retina and they can tell if you have tears or detachments of your retina. And they go check all these blood vessels and make sure you have good, healthy blood vessels. Because if you don't have blood vessels to your neural receptors, you're not going to be seeing for very long. Yeah, and that is, so we regulate the amount of, and I'm going to talk about this probably next time because I'll probably kind of wrap up here for tonight. But you have cells. So have you ever had where you're outside and you're in really, really bright sunlight and then you come inside and you can't see anything, like the whole room's too dark, or vice versa, it's really dark and then you go into a bright room. So we have specialty cells that help to neurally adjust down the light in our eye. We try to physically adjust it with what we do with our pupils, but we also have cells that help to neurally like tone down what we see so we don't damage our receptors. So when you're out in really bright sun, it kind of cranks down the receptions, which is why when you come in, then everything's dark because your neural receptors got cranked down in their sensitivity. So I think with that, when they shine that into the back of your eye, you kind of kick off that effect, but not at first because it's really bright, but eventually your eyes will kind of adjust to that. And we'll talk about those cells that do that. All right, so let's just look at this picture, and I think we've kind of wrapped up our anatomy here, but here's our cornea. Here's our lens. We have some ligaments that attach into our lens, which are they're called ciliary ligaments, which are going to help to change the shape of the lens, and we'll talk about how we do that a little bit more next time. We know our sclera, our choroid pigmented layer, our inner neural layer. Our visual axis is going to this little circle right here. What is it? Fovea centralis. What's the area around it? Macula, and it's high in what? What receptors? Yes, but what's the receptors? Rods or cones? Cones, high in cones. What's this where our nerve comes into the back of our eye? Optic disc, and it is also our blind spot, right? This is our blind spot. And that's because you can see right there, I have no neural receptors right there where the blood vessels and the nerves come in. So this is our blind spot right here. All right, we're going to stop there. We will start with the iris next time on Thursday. We should be able to easily finish this chapter on Thursday, and then we'll be ready to start endocrine. We'll go really fast through endocrine because you guys pretty much got a little mini lecture on endocrine in lab today. How do you guys feel about what we went over in lab on endocrine?